He also hosts what's probably the coolest and weirdest field site on the Elkhorn River, a little field station there. So if you're ever up that way, check it out if you can. But prior to joining the UNO faculty in 2018, he completed his doctoral training in freshwater ecosystem ecology at the University of Georgia and was a postdoc researcher in the Stream and River Ecology Lab at The Ohio State University, where he studied connections between aquatic and terrestrial food webs. At UNO, his research focuses on the ecosystem ecology of streams and rivers in the Great Plains, including strategies to manage the multiple stressors that threaten freshwater ecosystems in the region, such as nutrient pollution and urbanization. And of course, some of you may also know David because he is an active participant in the Great Plains Streams Working Group and also collaborates with my lab up in the Niobrara River on our project there. So really excited to have you here today, David, and I'll let you take it away.
Yeah, thanks, Jess. Great to be here, and yeah, good to see some familiar faces. You're probably wondering why I didn't just drive over, but I am grateful for the ability to do this on Zoom. It kind of makes my schedule a little bit easier today, so I appreciate that. So I'm going to start sharing my screen here, and hopefully y'all can see that now. Yeah, so today I am going to tell a few stories about how I use what I'm terming big data here, although if there's any bioinformaticians in the room or remote sensors in the room, they're probably laughing at me for saying that. But how can we use big data to propel new insights in freshwater ecosystem science? And so as a stream ecologist, I kind of have to put up a figure that looks something like this, where I say something about how the stream is ruled by the valley and that they exist within a watershed. So it is important to remember that the way that a stream looks and operates is largely dictated by what's going on within the land that surrounds it. So in a watershed like this is a picture of a forested stream in the Little Tennessee watershed in North Carolina, where I did my dissertation research. And most of the streams there look something like this, pretty typical forest here, and then some nice boulders and things of that nature. But of course, when I moved here to Nebraska, it was a very different story in terms of what the watershed and how it's shaping the stream. So in this case, we're looking at a mixed land use stream in the Papillion Creek watershed here in Omaha, and it's a much different stream, and it potentially works in many different ways as well. One of the ways that we are changing the way streams are working is through shifting their chemistry. And specifically, I'll focus today on how we are changing the carbon, nitrogen, and phosphorus stoichiometry within watersheds. And one question I try to answer is, how will streams respond to that change in chemistry? So this figure here is showing the in teragrams per year nitrogen and phosphorus inputs to the biosphere that are driven by humans. And then if we look at how that is changing the chemistry of watersheds, this is a picture of how nitrogen and phosphorus are changing relative to land use that are listed over here, like agriculture. So if we look at agriculture in the watershed, we see that these warmer red tones here are where there's lots of agriculture, and there's more extremes in terms of more nitrogen or more phosphorus within stream water in these agricultural watersheds. So we're changing the stoichiometry of and the chemistry of the watershed. This is important in the context of the plants and animals that live within streams. All of these different animals that we know and love require a certain proportion of carbon, nitrogen, phosphorus. It kind of depends on where you are within this food web, But a good benchmark to think about is what we call a red field C to N to P, which is 106 to 16 to 1 carbon to nitrogen to phosphorus. So that will come up again a little later. So these characteristics of streams, and that they're situated within watersheds and they're experiencing these changes, really set them up to be a good testing ground for different global changes, including those changes to watershed stoichiometry, as well as interactions with things like the climate and changes to their flow regime. So if we're changing the chemistry of the watershed, and there's just more or less water depending on what time of year it is or the climate, how does that translate to the export of these nutrients and elements from watersheds to downstream and eventually the ocean? The other nice thing about this data, or thinking about a stream in this way, is that there's a lot of data out there on stream flow, as well as nutrient concentrations and carbon concentrations within streams. And these are just a few of the sources of data that I'll be talking about today. And this is kind of just a plug for them, just a shout out, if you will, to USGS and EPA and the National Ecological Observatory Network. But as stream ecologists, we're dealing with, as many researchers are, an unprecedented volume, variety, and velocity of data acquisition. And I think one of the biggest challenges that we face is how to thoughtfully engage in what I call data remixing. So all this data is out there, but how do we make any sense of what's going on? So today I'll be talking about 3 different ways that I think big data can inform freshwater ecosystem ecology. And these are just 3 that I picked because I happen to have some data that would kind of illustrate what I'm talking about, but there's certainly many, many other ways that we could use big data to really confront some of these problems that we face in freshwater ecosystem ecology. So one that might come to mind is understanding extreme events. So something like this, where what happens when we have a rare event. In many cases, you can't run out there and figure out what happened until after the fact. But if you've got big data and a monitoring program, then you can understand extreme events. Another one that I'll talk about is intra and interannual change. So how are we changing these systems throughout the year in response to seasons, as well as from year to year? So this green line might represent a given year, and then the next year might be totally different. Without big data, we really can't detect that, and it makes it a lot easier to do so. And the third way that I think that we can use big data in freshwater ecosystem ecology is to expand to really big scales and think about macro systems and how there are interactions across different scales from region to continent, as well as teleconnections that are driving some of the processes that we see within streams and rivers. So these 3 stories I'll outline here. The first one is looking at an extreme event, specifically one that we might all remember from 2019, where I'll show you some data from pre and post-flood nutrient fluxes within a Great Plains River network. The second one is looking at intra and interannual change, where we're using high frequency monitoring at Glacier Creek Preserve to understand how carbon is exported downstream from a mixed land use watershed. And then the third is looking at these cross-scale interactions or macro systems. And this is a new project I'm working on where we're exploring the continental scale role of fine particles in riverine material fluxes. All right, so let's get into this first one. So in case you did not live here in 2019, throughout the Midwest, 2019 was not your average year for precipitation. this map is showing the precipitation anomalies or departures from average for the calendar year of 2019. You can see in some places just south of us in the Great Plains, we're over 20 inches above normal. We were more in the 8 to 12 inch range, I think, here in Nebraska. But regardless, tons of rain that year, tons of precipitation. Of course, all that water falling on the ground had to go somewhere. And so it was also not your average year for stream flow. This is a picture that Jess Corman actually took of the Missouri River near Omaha in March of 2019 at the sort of the peak of the flood that year. And so you can imagine that there's all kinds of stuff floating in this water. It's not just water. And one question that we're interested in finding out is, okay, what exactly happened during this flood in terms of what is carried within the water? So one way that I'll attempt to do this is to look at concentration discharge relationships, because they can often reveal biological versus hydrological controls on stream carbon, nitrogen, and phosphorus fluxes. And I think it's helpful to think about it in terms of this figure here that I took from this paper, where we're looking at discharge on the x-axis, and then concentration of could be carbon, nitrogen, or phosphorus on the y-axis, and then looking at how that concentration changes as a function of discharge. In many cases, it's a power law relationship where you get this nice increase in concentration as a function of discharge. The opposite could also be true. You can see a decrease in the concentration with discharge. And this is related to two different processes. One would be hydrological export, where basically when there's more water, you get more stuff floating within the water, and then that flows downstream. The opposite could also be true. You could have more water diluting out whatever is dissolved within the stream or river. And so these are two different processes that could be occurring depending on what you're looking at. Also important to note that at low flows, you're going to have potentially higher biogeochemical activity, more retention of, say, particles and other constituents within the river, as well as its removal before it moves downstream. And then the opposite extreme, we tend to see hydrological export at high flows, as you might expect. So with more water, more of it's getting flushed downstream and eventually to larger rivers and oceans. So if we look at how we can understand the flood event that happened in the Platte River network in 2019, our overarching question here is, how did the 2019 flood event affect the flux of carbon, nitrogen, and phosphorus from this Great Plains River network, where we saw this anomalous rainfall? And specifically, we're talking about the bomb cyclone event that happened in March of 2019, where there's kind of this perfect storm of snow, freezing, and then precipitation that led to this historic flood event. Luckily, U.S. Geological Survey collects data on a daily time step from 3 sites within this river network that also happen to represent a nice hydrologic gradient. And by that, I mean, we're looking at a smaller creek here, Maple Creek in Nickerson. And then we get into larger rivers, the Elkhorn River in Waterloo, and then the Platte River in, is it Louisville, here in Nebraska. So 3 sites of increasing size where we've got some good data where we could try to link up what happened during this flood event. So I specifically looked at the data that was collected by USGS. Again, we retrieved stream flow data, and then total organic carbon, total nitrogen, and total phosphorus concentrations that were collected from grab samples from Maple Creek, Elkhorn River, and the Platte River. And the nice thing about this is that there's this really great package called data retrieval out there that allows you to use our software and essentially connect to USGS server and then link all this into your R console, and then you can work with the data really easily. So it's a really nice package that's great for lab modules as well as doing research like this. So I went back to the earliest date where they had reliable grab samples of carbon, nitrogen, and phosphorus, which was 2008. Started in that water year in October, and then went through October of 2020. So this is a total of 4,570 days. With that data in hand, we have the grab sample data, the water chemistry, and then we have the daily stream flow data. We were able to use another package called LoadFlex, which allows us to model the concentration and the fluxes of carbon, nitrogen, and phosphorus during this time period. Basically, this uses a composite model that is looking at a linear regression, so that discharge and concentration relationship, and then also uses interpolation to fill in the blank spots between grab samples. So in this way, we've got a really nice time series of carbon, nitrogen, phosphorus within these 3 sites that's also linked up with stream flow. All right, so let's get into the data. And what I'm showing you here are the concentration discharge relationships for carbon, which is shown in black, nitrogen, which is shown in blue, and then phosphorus, which is shown in red. So the concentrations on the y-axis, and then the discharges on the x-axis. The 3 panels are splitting it up into the 3 different sites. So we have Maple Creek, which is the smallest site, Elkhorn, which is in the middle, and the Platte River, which is the largest. And if you remember back to this figure here, where I showed you the typical relationships between concentration and discharge, what we see is that there's some hydrological export going on within these 3 sites, at least for these 3 constituents, the total organic carbon, total nitrogen, and total phosphorus. A few other things that I want to point out here. One is that the slopes of these lines for carbon and phosphorus tend to be more similar in all 3 sites. The slope is a little bit lower for nitrogen, especially in Maple Creek and the Elkhorn. And the slopes tend to be higher in Maple Creek compared to the Elkhorn and the Platte. And this will become clear why I pointed that out, hopefully here in a second. So if we take that information and we know with some certainty that there's some hydrological export going on, and that with higher flows, you're getting higher concentrations of carbon, nitrogen, and phosphorus, what we can do is calculate a flux, which is simply multiplying the discharge by the concentration. And that's basically going to tell you how much stuff is floating downstream. Technical term, I know. So what I've done here is I've parsed out the total amount of water that is flowing through each of these sites on an annual basis, and then converted that into a proportion of the daily flow that's happening on any given day. So for example, in Maple Creek, we've got 11% of the annual flow happened on March 14th, 2019, when we had that flood event. I can do the same thing for carbon, nitrogen, or phosphorus flux and look at this in terms of what happens on an annual basis, and then get a proportion of that happened on a daily basis. So what I'm showing you here is how much carbon, nitrogen, or phosphorus was exported on a given day as a proportion of the entire year. So I've got that for Maple Creek here, Elkhorn here, and the Platte River. And then I did it for carbon, nitrogen, and phosphorus again. And this solid line is showing you a 1-to-1 relationship. So if basically the amount of water and the proportion of carbon, nitrogen, and phosphorus was the same, it would match up with that line. But what this is showing is that in general, across all 3 sites, we see a higher proportion of carbon, nitrogen, and phosphorus per proportion of water that is leaving the watershed. Another way to think about it is on this flood event here in 2019, 40% of the annual load of carbon, nitrogen, phosphorus, or annual flux, I should say, of carbon, nitrogen, of carbon went left on that day. Versus if we look at the Elkhorn and the Platte, that's a much smaller proportion that is leaving even during these high flow events. So this suggests that the smaller site, Maple Creek, is more dynamic in how it is, how fluxes of these nutrients are leaving the system compared to these larger rivers. And these data are like right off the press here. So I'm being a little bold in showing them to you because I haven't completely thought through what's going on here. So with that caveat, I am going to point out that if we look at the stoichiometry of, or the ratio of how nitrogen and phosphorus are leaving all three of these sites, what we see is that there's a really strong coupling between nitrogen flux and phosphorus flux, regardless of what flow is or apparently land use. So there's a remarkably consistent relationship between the amount of nitrogen that's coming out of each of these different sites, no matter what is going on. And I'll also point out that the slope of this line for each of these different sites is remarkably close to the Redfield and the Peat. So I'm not exactly sure what's going on here, but definitely something that we'll be exploring here in the future. Okay. So to sum up this story, what we're seeing is that for concentration discharge relationships, total carbon, nitrogen, and phosphorus suggests that each is transported during high flow events, and we're seeing hydrological export. Smaller streams transport greater proportions of carbon, nitrogen, and phosphorus during high flows. They seem to be more biogeochemically and hydrologically dynamic compared to larger rivers. And this maybe isn't too surprising, but we've got the data now to really confront what might be happening. And then this pattern, the TN versus TP flux stoichiometry, is consistent across the network, regardless of high flows and intensive land use. So something to explore in the near future. All right, for the second story, I'm going to shift gears a little bit and think less about rare events and more about sampling frequently at one site and using that information to understand intra and interannual change or lack thereof. So now we're taking a trip over to Omaha. This is Glacier Creek Preserve, and you can see that there is a creek here. It's very small, kind of hidden by all these grasses. It's a restored prairie that is managed by the university and the Department of Biology here at UNO. And it's a great resource, super cool site. If you ever have a chance to come visit, please do, and I'd be happy to give you a tour. I want to acknowledge that this work is largely driven by a really great group of folks here at UNO, Andy Miller in chemistry, Ashley Deer in geology, geography, and Tracy Coleman, who's the outreach specialist on site and also does all the maintenance of the sensors that we have out. And then Tom Bragg and Barbie Hayes are really the drivers who started Glacier Creek Preserve in the 70s and have been really having a great vision for what could happen there in terms of teaching and research. So it's a great resource, and I'm really excited and fortunate to be able to work there. So we have got this kind of overarching question as a group of how watershed scale restoration is affecting stream water quality. And I'm specifically interested in how it's affecting carbon transport, storage, and transformation within nearby streams. We're kind of using this as a testing bed to understand the effects of restoration at a watershed scale on these processes. And this is important from the context of river ecosystems because they do transport, transform, and store tons, literally tons of carbon. So this is a figure that's fairly old now. Some of these numbers might be updated slightly, but basically streams, rivers, and lakes receive 2.7 petagrams of carbon. A lot of that is outgassed as CO2 to the atmosphere. Some of it is stored, and then the remainder is transported downstream to the ocean. So we have got active participants in the global carbon cycle. And it's important to understand each of these different arrows and what's going on. An extension of this idea that's come out in recent years is this idea of the pulse shunt concept, which is surprisingly hard to say that I'm saying it out loud. But this is the idea that streams are likely operating on a continuum of passive carbon transport versus active carbon processing. And this figure is showing that here. We're at low flows or low discharge. Perhaps these streams are more active, and then at high discharge, it's more of a passive pipe. Anything that's dissolved or transported within the stream is just going straight downstream. It's not being actively processed by microbes or organisms living within the stream. So to kind of revise this question a little bit further, I am interested in understanding how and when do these pulse shunt carbon exports occur within this mixed land use watershed. So one way we can measure and understand these pulse shunt dynamics is to look at carbon spiraling. And I don't know if Steve Thomas is here or not, but he really pioneered this along with several other folks. So I should probably be asking him about what's going on here. But basically what carbon spiraling allows you to do is look at the ratio or the difference between the export or the flux, so discharge times the concentration of carbon within the stream, divided by the respiration or processing of that carbon. And so we can think about this in two different scenarios, kind of two different extremes. This is a rain event that I managed to get a picture of in Elmwood Creek, which is the stream that's nearby UNO's campus. And this situation here, after we received about an inch of rain, is potentially more of a passive pipe situation where we have low mineralization rates, low respiration. That is, the high discharge means that we have really long spiraling lengths. So carbon is coming in and it's being immediately shunted downstream. Contrast that to 4 days later when we're back to base flow. This is more of your active reactor situation where you have high mineralization rates, lower discharge, resulting in short spiraling. So to get this question, we're using high frequency monitoring in Glacier Creek. This is a picture of the watershed and the monitoring location, which is basically at the end of the section of the stream where there is substantial vegetation restoration. So this box here represents the area where the tallgrass prairie has been restored. Most of the land surrounding that is still in agricultural production, either corn or soybeans, depending on the year. So we've got this YSI XO2 out in the stream. It's been out there since 2017. It's equipped with dissolved oxygen, fluorescent dissolved organic matter, and turbidity sensors that measure each of those things, plus a few others, every 15 minutes. So over the span of now almost, well, more than 4 years, almost 4.5 years, we're at greater than 100,000 points for each variable that we're collecting. What we can then do is crunch all these numbers and model whole stream metabolism to estimate heterotrophic respiration rates, or that carbon processing, as well as total organic carbon concentrations from the turbidity and fluorescent dissolved organic matter sensors. The combination of these data allow us to estimate at high frequencies, daily time steps, carbon spiraling, both the mineralization velocity, or VFOC, and the spiraling length, or S. So what are we looking at here? Again, we're looking at intra and interannual change. And by and large, interestingly enough, Glacier Creek seems to show typical spring peaks in gross primary production and respiration. I'm showing you this just to highlight that basically when you have light in the stream, primary production turns on, so photosynthesis turns on, and that's also driving heterotrophic respiration in the stream. So primary production is shown on the green dots up here. Respiration is shown in the orange dots down here. The difference between the two, or net ecosystem production, is shown over here on this panel. So by and large, we're seeing spring peaks each year in primary production, and that's driving respiration rates in the stream. But what are we seeing in terms of carbon spiraling? So this graph is showing mineralization velocity, or that carbon processing rate, essentially, and then this carbon spiraling on this graph here. And the pattern that I'm noticing, so I've binned everything by month, and then this is all the months of January that we have. So each day that we have data in January, in 2017, 2018, and so on, is shown with the gray dots here. And the major pattern is that there's a similar spring peak in mineralization velocity, and then there's a dip in spiraling lengths in the spring as well. So this is mirroring what's happening with ecosystem metabolism, suggesting that's really driving carbon spiraling within Glacier Creek. We can also look at this in terms of discharge, similar to the concentration discharge relationships that we looked at before. And this graph is showing you discharge within the creek, and then carbon spiraling length on the y-axis. This blob of gray dots is all the daily time steps of carbon spiraling that we have for Glacier Creek. And then these open squares, open circles, and open triangles are various literature values that I pulled from these papers down here. And what I want you to notice from this is that carbon spiraling is a dynamic process that's varying quite a bit within this within Glacier Creek. But the majority of the data, which are found in this center or centroid of this kind of blob here, if you will, is comparable or in line with streams of similar discharge. But again, it's a dynamic process in that sometimes the carbon spiraling is comparable to larger rivers. Some of these are like the Colorado River, some of or even larger than that. And that's about 4% of the time. And then the other extreme is that sometimes that carbon is being processed before it leaves the Glacier Creek itself, in that the carbon spiral is shorter than the reach length in about 9% of the observations. But that means that most of the time it's somewhere in the middle. And so it really is this continuum of carbon processing that we're seeing within Glacier Creek. So you might be wondering, well, when does Glacier Creek act like a river? The maximum carbon spiraling we observed happened to occur on the day before this video was taken. So it wasn't quite this extreme. In fact, we weren't able to measure ecosystem metabolism. It's really hard to measure in conditions like this. But this is to give you an idea of what the stream looked like during that flood. Yeah, so for some context, this stream is normally less than half a meter across, and here it's got to be 2 or 3 meters. So it's really responding rapidly and in a dynamic way to this event. So to sum up what's going on in Glacier Creek in terms of carbon processing and carbon spiraling, we see that seasonal patterns are driving mineralization and carbon spiraling lengths, and they mirror ecosystem metabolism. We're seeing some evidence for this pulse shunt export of carbon during high flows. Active processing is occurring during lower flows, and both of these extremes occur infrequently within the stream. One question that I'm hoping to answer in the future is, how do larger streams respond across their own gradients of flow, across their flow regimes? Do we see a similar pattern in terms of this dynamic change in carbon spiraling from low flows to high flows? That's going to be really challenging to figure out, and may not ever get to it, but something to think about. All right, so the third story I'm going to tell you about is looking at cross-scale interactions and how we can use big data to understand some of these processes at a continental scale. And specifically going to talk about fine particles. In this new project I'm working on in collaboration with John Benstead and Ariel Sjogren, as well as the National Ecological Observatory Network, where we're going to investigate the character and flux of fine particles from wadable streams across the United States. So to back up for a second and define what I mean by stream particles, another word or term that we use is ceston. This is basically anything that's not dissolved that's still carried within the water of a stream or a river. And we know a little bit about the composition of what these particles are. It's probably a blend of inorganic particles from soils, as well as organic detritus, and then living and dead microbes, including algae. And it serves several important functions within stream and river networks, including linking that watershed, the terrestrial landscape, to the aquatic ecosystem. It's also an important flux of carbon, nitrogen, and phosphorus, as I showed before in the Platte River network. And it's an important food web component. So lots of particle feeders like freshwater mussels, black flies, and other things that we know and love depend on these particles for food. Now, much like dissolved constituents within streams and rivers, we know that there are different fates or potential fates of these particles. They could be buried, they could be processed and converted from an organic form into inorganic, could be carbon dioxide, could be something else. And then some of it might be transported downstream, as we saw previously. It's also really key to understand this compartment in a changing world. These are some more pictures from the flooding that occurred in 2019. And you can see that the Elkhorn and the Missouri River are just full of stuff that makes it look like chocolate milk, again, for lack of a better term. This is a sample that we were able to collect from the Elkhorn, and you can see that even in this opaque bottle, it's really super cloudy and full of particles. So in a changing world where flow regimes are changing and these episodic events are becoming more frequent, what's going to happen in terms of particles, their transport, and their processing. So to get at this, we're looking at 22 streams that are managed by NEON across 14 different domains, which are shown here from Alaska to Puerto Rico. There are some data that have been collected previously from 2014 to 2021. But this is usually at a kind of a seasonal time scale. So they collect 3 times a year in the spring, summer, and fall, basically during the growing season. And we are increasing this frequency to bi-weekly, essentially. So in most of these sites, we'll be getting bi-weekly ceston samples, grab samples from the water. And then we're going to be able to link this up with other data that are collected by NEON. So we did a little bit of exploring of the existing data. I want to show you that here, just because I think it's fascinating in that across the NEON wadable streams, if we look at the ceston carbon and nutrient content, there's some differences in terms of carbon to nitrogen, which is shown on this panel here, and then carbon to phosphorus, which is shown in this panel here. In terms of carbon to nitrogen in the ceston, they tend to be more tightly coupled, and by that I mean that they're following this line in a much tighter pattern compared to carbon to phosphorus. And they're also kind of tracking, again, red field C to N. So across the continent, if you pull out some ceston, the concentration or the ratio of carbon to nitrogen in these samples is right at about 13 to 1. Of course, there's a lot more variability in carbon to phosphorus, which is interesting, and we're not exactly sure what's going on here. That's something that we're hoping to find out. It could be maybe a geologic signal, maybe differences in phosphorus availability within the watershed itself. Could be differences in carbon processing, not exactly sure at this stage. We did look at carbon to phosphorus in ceston across the United States. This is just box plots of ceston C to P in the different domains. And you can see that they're quite variable depending on which sites or which domains we're looking at, but there don't seem to be any detectable spatial patterns, at least looking at it in this kind of coarse way. We do see that ceston C to P decreases with watershed size, suggesting that in smaller streams, like these up here, there may be higher carbon to phosphorus in the ceston, reflecting either differences in the source of that ceston, or perhaps differences in their processing. So perhaps in these larger watersheds, which tend to have higher or larger rivers within them, perhaps these are more processed. There could also be some differences in climate. This is Sycamore Creek in Arizona. It's a desert stream. This is the Arickaree River in Colorado. It's a prairie stream, so there could be some differences there as well, just based on vegetation. One thing that we're excited about doing is linking up the ceston data with co-located sensors within each of these streams. So I'm showing you some data here that's collected on a 1 minute time scale from each of these sites. And we expect that with these sensors, we can link up stream flow, the grab samples of ceston, and related metrics like turbidity and chlorophyll that might be driving some of the characteristics of the ceston that we're collecting. And we can see that there are some potential differences across different sites that might be interesting to explore. I'm showing you data here from the Arickaree River in Colorado, which are shown in the blue dots here. This is looking at a time series of turbidity throughout a year, April to April. And then the red dots are showing Walker Branch, which is a forested stream in the Appalachian Mountains in Tennessee. And there's some pretty big differences in terms of when turbidity peaks in these two different sites. In Walker Branch, we're seeing a turbidity spike in April of each year, potentially coinciding with when primary production peaks. And we're definitely seeing a peak in turbidity in during the growing season in the Arickaree River, which seems to line up with chlorophyll A. So we've got chlorophyll A on the x-axis here and turbidity on the y-axis in this one, on this panel here. And there's a strong relationship between chlorophyll and turbidity, suggesting that this is being driven by primary production in the Arickaree River during the growing season. Another aspect of the project that we're working on is exploring how particles are processed and transported differently within different sections of a river network. And luckily, Neon has a hydrologic gradient built into their sites. And so the folks in Alabama, Ariel and John, will be taking the lead on looking at these particles in Mayfield Creek, the Black Warrior River, which is downstream, and then the Tom Bigby River down here. So we're looking at particles as how they change from these forested small watersheds to much larger rivers where we have to, they will have to go out and sample on a boat or using these kind of moored buoys to sample the ceston and other characteristics of the river. So just to quickly summarize, this particular project here, I'm really excited about the opportunity to study the interactions among the climate and geological setting and looking at those cross-scale interactions. We are expecting to see that Ceston will be an integrated signal of terrestrial and aquatic processing that will tell us a lot about what's going on within these streams and rivers. And then we also are excited about the opportunity to couple high resolution sensors with this large spatial scale and grab samples to reveal these cross-scale interactions and teleconnections. So I'll leave it there, and I'm going to show this picture from the Niobrara River just to illustrate that I'm not always stuck behind my computer analyzing these big data. I do get out and do field work. So if this sounds interesting to you and you're looking to do a master's degree or work with me in some way, please do reach out. We have a lot of fun. And I'll also shamelessly plug, is hiring an ecologist position right now. So if that would be of interest to you, please apply or get the word out. So we are hiring. But with that, I'll take any questions. I hopefully have some time to do that. And thanks very much for listening.
Awesome. Thanks, David. That was a great talk. We'll give people a chance to ask questions. Remember, please submit them in the chat, or I think if you'd like to raise your hand, I can call on you too. But I think I'm going to launch off with a question, which is, in this new project, looking at stream stoichiometry, what processes, like I'm assuming climate is one that you're thinking about with the teleconnections?
Yeah, with teleconnections, the primary example of that is like El Nino and those sorts of processes, which are happening at much larger scale than even the continent here. So certainly climate is one that we're thinking about, but we are also spinning the hydrologic connections as a potential teleconnection. So this upstream to downstream connectivity that occurs within a river network, we're arguing is also a teleconnection, because we're dealing with something that's happening upstream and having an effect on something that's going on downstream. And so that's one teleconnection that we're thinking about as well.
Yeah. So sort of expanding the river continuum.
Yes.
On some ideas.
Right.
Yeah. Yeah. Cool. Waiting on some folks. Come on. I know you all have good questions. Okay, there's one in here. I won't entirely use up your time, David. Okay, so have a question in the chat. First one is, because these are bigger data problems that are getting bigger, do you see a move away from R to tools like TensorFlow or Cafe or CAF? And please correct me if I've mispronounced that.
Yeah, that's a good question. And I think it is going to show how what we're trained on largely dictates what we use and think is useful. So I'm not familiar with either one of those things, so that might answer your question right off the bat. But certainly, I think there's a lot of tools coming online, which I think will help us even more than what R has been able to do. So I just happen to use R because that's what I'm familiar with. And I do think that as we get further into this, there's going to be a need for programming to really streamline some of this stuff. so that you're not, we're not spending all of our time managing the data in something archaic like R, which is sad that's becoming that way. But yeah, I don't know if that answers your question or not, but.
So there's a second part to the question, and I'm going to ask a first sort of an intro question to that, which is, you talked about using different sensors. What parameters do sensors work well for, and what parameters or what parameter would you love to have a sensor for that maybe isn't quite working at the moment or isn't available yet?
Yeah. So yeah, it's amazing what technological leaps we've had just with dissolved oxygen in the last 10 years. So the switch from basically a polarographic version to one that's an optical sensor has really increased the quality of those data. And so I think we're pretty good at measuring things like dissolved oxygen now. Turbidity is pretty good, I think. Stream flow we're pretty good at. The things where we're basically just measuring, stream flow, you can measure pressure or something like that, and it's fairly simple to do that. So applying, maybe technology that's used for other things like weather monitoring to other things. But yeah, I think certainly with any sensor, there's going to be problems. I know with some of the nitrate sensors, at least they've got optical ones now, but they didn't used to, and there's lots of problems with drift with those. And so with all these sensors, part of the challenge is not only figuring out what's going on and what's real, but also, okay, how do we deal with outliers that are probably from instrument error and not maybe process or observation error, if that makes sense. But for the second part of that question, I would think having a nitrate sensor, an optical nitrate sensor, would be great. We're trying to get one for Glacier Creek. And there's some potentially really interesting things that we could get at with having that high frequency nitrate. I think that would be really nice to have.
Yeah. And then the follow-up question was thinking about tools like snapshot hyperspectral imaging or Raman spectroscopy. Where are they in terms of using them for stream chemistry?
Yeah, that's a good question. I'm not entirely familiar with, is that like a remote sensing type of technology?
Yeah, and I think that the key there is that I think is it Kate Chapman? Anyway, I think there is someone in the audience who knows a lot about chemistry that should talk to you and I, because I'm interested in this too, in terms of getting some more sensors out.
Yeah. I know John Chalice at Creighton, who's down the road here, there's a lot with remote sensing and carbon. And he was the one who suggested getting the FDOM sensor for Glacier Creek. So I'm super grateful for that. But yeah, I think it's a little easier to do in larger rivers, I would guess, where you can actually see it. If you looked at that picture of Glacier Creek that I showed, it's completely opaque because it's covered by grasses. And so I think that's a huge challenge that we're facing with smaller streams, and kind of figuring out, okay, how much water is actually there and where is the stream exactly? You can kind of get that with the digital elevation models, and we're getting a lot better at that. But in terms of measuring something like chlorophyll in a small stream from a satellite, I think we're a long ways from being able to do that. Perhaps you could do something with a drone.
Yep.
But even that might be hard. Like at that picture of at Coweta that I showed, they're just covered by rhododendron. So it'd be hard to fly a drone through there.
Yeah. Okay, so I have another question, which is what role does big data play in coupling the analysis of streams with other aquatic ecosystems? How do we couple the different spatial temporal scales in which nutrients are processed in multiple types of aquatic systems?
Yeah, that's a great question. Yeah, I think too often, we don't necessarily think about, we get specialized. I'm a stream ecologist, so I like to think about streams, but certainly there's folks who are doing very similar work in lakes, estuaries, wetlands, and they're all within watersheds, ostensibly. So there certainly could be some cross-fertilization there that I think would be really helpful to understand. Okay, if we're looking at the Platte River network, there's a bunch of reservoirs, or the Missouri, there's a bunch of reservoirs on it, So from the standpoint of flux of carbon, nitrogen, and phosphorus, I can understand what's going through the parts of the river that are flowing, but then once it hits the dam, what happens then? I think that's a really interesting question, and I'm not sure that the right people are talking to each other about that at this point. But
right, there was another question which you effectively just answered, which is, have you considered adding discharge and nutrient gauges across the state to examine gradients in land use, spiraling lengths, as well as nutrient flux relationships? Yeah, we've both thought about that.
Great.
If anyone wants to give us money. And then, so I have another question here, just a clarifying question, which is about the Redfield ratio and why you're comparing with Redfield ratio when you're looking at these concentration of flux data.
Yeah, that's a great question. I kind of glossed over that at the beginning, but it's the Redfield ratio refers to this sort of canonical concept that in the oceans, the concentration of carbon, nitrogen, phosphorus in the water is the same as the concentration that's contained within algae, suggesting that there's this kind of reciprocal requirement versus availability, if you will, so that they're driving each other. And yeah, I guess I was just kind of geeking out and putting that in there. But it is interesting in that it's often used as kind of a benchmark for what the requirement of different organisms are. And so in many cases, we can sort of use it to say, well, there's a threshold between where carbon, nitrogen, phosphorus might be more or less limiting depending on where you are, below or above Redfield ratio. And so it's interesting that a lot of these things are falling out along that line, and suggesting that we are seeing kind of a biological signal in the stoichiometry of some of these things.
Yeah, I mean the data that you threw up there for the nitrogen and phosphorus from the three systems and around Omaha was pretty crazy. I mean, that's, yeah, remarkably consistent.
Yeah.
Yeah.
And I don't know what's going on there. Because if you look at concentration, it's more of a scatter shot, but as soon as you account for discharge, that line just straightens right out. So I
wonder if it's just because it's all being so hidden because it's a log.
Yeah, that could be too.
Yeah. And it's, is it the signal of the watershed or is it the signal of river processing?
Right. So I would hazard to guess that in a different watershed, say one of those agricultural ones that had the super high N compared to P, that you would get a different signal. So.
Well, John has turned on his video, which means I think we're supposed to let you go.
Okay.
Yeah, I hate to break up the party, but I think just to be respectful of everyone's time, we've done a good hour here, and great talk, David. Thanks a lot.
Yeah.
And a good discussion too. And so I guess, yeah, everyone that's left, maybe if we can just give some sort of Zoom applause to David, and yeah, just thanks again.
Thanks, everyone. Really great to be with you today.
Yeah, thanks, David. And in the comments too, lots of people said thanks.
Okay, thanks everyone. Hope to see everyone next week in person or on Zoom with Dr. Mike Chamberlain. We'll see you next week.